Hello, everyone. Welcome to the Research in Immunology Professional Community webinar, the best of ISHLT conference abstracts. My name is Eric Morrell. I'll be one of your moderators, along with Dr. Billia, who's our other moderator. So we have two speakers today, and then our third speaker unfortunately couldn't make it, and so we'll have a recording of that individual's talk. Each speaker will have a 15-minute presentation, and then we'll have 5 minutes at the end where we can unmute people. Just raise your question, raise your hand, and then we can ask questions. You can also type them into the chat as we go along. And so without further ado, I'd like to present our first speaker, Julie Buchwald, for the University of Massachusetts, who was the 2024 Early Career Scientist Award winner. And the title of her talk is Optimizing Hearts for Transplantation Using Small Interfering RNA-Based Therapies During Ex Vivo Machine Perfusion. Thank you.
Okay, thank you so much, and good afternoon, everyone, and thank you for having me. I'm excited to share with you all my work during this webinar. And so organ transplantation is uniquely suited for therapeutic intervention. 10 to 30% of heart transplant recipients develop primary graft dysfunction, partially due to ischemia reperfusion injury, or IRI, contributing to significant morbidity and mortality, and for which there are no effective therapies. We believe that harnessing the platform of ex vivo heart perfusion, or EVHP, to target IRI using siRNA-based therapies would allow for the rehabilitation and ultimate transplant of hearts currently deemed untransplantable. Advantages of EVHP in general are well known and listed on the left, and includes the fact that it dynamically preserves organs outside of the body at physiologic conditions, allows for real-time monitoring of organ function to assess suitability of transplant, does not require costly systemic therapy due to the treatment of the organ in isolation, and lacks off-target and off-organ adverse effects with a proven safety profile given its current clinical use. EVHP is also a uniquely appropriate time to administer siRNA-based therapies because siRNA harnesses endogenous mechanisms to downregulate targeted genes without the use of viral transfection agents in already immunocompromised hosts. With siRNA, there is no concern for insertional mutagenesis, tissue tropism, or gene size intended for delivery. And finally, siRNA avoids permanent gene knockdown or knockout, which may prevent toxicities associated with prolonged homeostatic signaling repression. The introduction of EVHP into clinical practice has increased the number of heart transplants performed each year in general, including those from DBD and DCD donors. However, there remains no clinical therapeutic in use aimed at mitigating IRI, so IRI persists. As such, there is currently no strategy to rehabilitate and optimize donor hearts for transplantation using EVHP alone. And this accounts for the discard of nearly 2/3 of procured hearts, partially due to this IRI-induced damage. This persistent and unresolved clinical problem opens doors then for new strategies such as ours, by which we can leverage the platform of EVHP to specifically target IRI pathways using siRNA-based therapies to not only protect, but in fact enhance the pool of transplantable hearts. The mechanism of siRNA involves delivery, where the siRNA duplex enters the cell, incorporates with a protein called RISC, then base pairs with the target mRNA of interest for cleavage and degradation, thus preventing harmful protein production. There are currently 6 FDA-approved siRNA drugs with many more in clinical trials, where a single systemic injection supports 3 to 6 months of efficacy, overlapping with the critical period of primary graft dysfunction. And we have systematically defined siRNA chemical modifications in the Kerckhof lab that enable efficient heart silencing, all of which stabilize our siRNA such that it requires no formulation or delivery vector. Pictured here is a schematic of siRNA conjugated to docosahexaenoic acid, or DCA, a naturally existing 22-carbon saturated fatty acid that we discovered enables heart silencing efficacy of 70 to 80% knockdown for over 1 month without toxicity. One of the advantages of siRNA drugs is that they are informational, where their delivery and potency stem from their chemical structure, or dianophore, whereas their target is defined by the base sequence, or pharmacophore. And so both features can be optimized separately, making siRNA drugs more programmable than traditional small molecules. And the DCA conjugate class is currently in phase 1 clinical trials under an FDA Fast Track designation for the treatment of preeclampsia, reflecting its efficacy and safety. And so then the use of DCA conjugated siRNA, therefore, is a realistic therapeutic option for gene modulation during EVHP. Our exploration into the feasibility of siRNA delivery and silencing during EVHP was launched using our porcine EVHP model in the Robbie and Pearson labs at MGH. Our model siRNA targets the inflammatory mediator JAK1, which has been previously published by our lab to robustly knock down porcine, human, and rodent JAK1 mRNA and protein, thus facilitating multi-species studies to assess platform efficacy. JAK1 siRNA is conjugated to DCA, and it's also labeled with a Cy3 fluorophore to enable visualization via fluorescence microscopy. DCA siRNA was added at a concentration of 1 micromolar in circulating perfusate, and total ventricular biopsies were taken over time for 6 hours of normothermic EVHP. At baseline, there is no Cy3 signal, but over time, we start to see Cy3 fluorescence within heart tissue in as early as 1 hour after EVHP. And these images come from biopsies taken over time, and thus they're from the outer epicardium, as you can see here. And we can see that then the DCA siRNA broadly distributes throughout both ventricles over time. We also see broad DCA siRNA distribution in biopsies from full-thickness myocardium taken at the end of the 6 hours of perfusion, supporting EVHP as a platform for robust, uniform siRNA distribution throughout all cardiac layers. We then wanted to assess JAK1 target knockdown. We see JAK1 mRNA levels increase over time in biopsies taken during perfusion, with JAK1 expression remaining consistently elevated across heart regions after 6 hours. This then prompted us to ask, is an increase in JAK1 mRNA expression due to the process of EVHP itself, and the JAK1 siRNA unable to suppress this increase over time, or is the JAK1 siRNA in fact dampening JAK1 expression levels compared to untreated organs over time? To answer the second part of this question, we turn to our collaborators in the Tessier lab, which houses a robust and reproducible rat normothermic EVHP setup in which the aorta is cannulated for Langendorf perfusion. And this model pictured here has allowed us to dissect the role of DCA siRNA in EVHP, examining distribution across cardiac cell types and efficacy at the whole organ level. Fluorescence microscopy of rat hearts perfused with DCA siRNA demonstrates uniform and robust siRNA distribution throughout the organ, like results seen with the porcine EVHP model. On the left here are control hearts, which were perfused for 6 hours without siRNA. In the middle are hearts perfused with unconjugated siRNA, and on the right are hearts perfused with DCA siRNA. We see uniform Cy3 signal throughout cardiac tissue with the DCA conjugated siRNA treated hearts across multiple rounds of perfusions, with the DCA conjugate enhancing siRNA delivery by 3.5-fold relative to unconjugated controls, emphasizing that EVHP serves as a platform for reproducible and whole organ treatment. We then wanted to characterize the cell-specific uptake of DCA siRNA during EVHP, specifically capturing uptake in cardiomyocytes and endothelial cells, as these are the predominant cardiac cell types affected by IRI. In the top panel, we can see untreated control hearts where DAPI stands for cell nuclei. Cy3 is coming from the labeled siRNA, MYH6 stands for cardiomyocytes, and wheat-germ agglutinin stands for plasma membranes, both externally and internally. In untreated hearts, we can see no Cy3 signal as expected, whereas in the unconjugated ex vivo treated hearts, we begin to see Cy3 signal. And then finally, the bottom 2 rows demonstrate the boost in Cy3 signal originating from the DCA conjugated siRNA treated rat hearts in red, where we can zoom in and see productive uptake of the siRNA treated rat hearts in red, and that captures the Cy3 siRNA in the intracellular endosomal compartments highlighted by these white arrows. And this is a necessary step in the productive internalization of siRNA into cells for robust downstream gene silencing. We can perform similar immunofluorescence staining for endothelial cells treated with CD31 in purple in smooth muscle cells, labeled with alpha SMA in green, where again, we see the benefit of delivering DCA conjugated siRNA during EVHP to robustly target endothelial cells compared to the unconjugated siRNA control hearts. If we zoom in further, we can then see that DCA conjugated siRNA administered during rat EVHP targets all layers of a vessel, including the intimal layer composed of endothelial cells, the medial layer composed of smooth muscle cells, and the external layer composed of other cell types such as macrophages and fibroblasts, suggesting that EVHP serves as a platform for not only whole organ treatment, but also whole vessel treatment as well. And this trend in robust cardiomyocyte uptake of DCA conjugated siRNA administered during EVHP is also seen following 6 hours of normothermic perfusion of pig hearts, as well as human hearts, where we can see no Cy3 signal here coming from the time 0 biopsies and a strong Cy3 DCA siRNA signal at 6 hours post-perfusion, captured uniformly in both the left and right ventricles of both the pig and the human hearts. And finally, we can see that DCA siRNA delivered during pig EVHP enables robust distribution to endothelial cells seen here, especially captured in these magnified images, as well as in human hearts perfused with the DCA siRNA. And so robust Cy3 signal from cardiomyocytes and vessels after 6 hours of human EVHP is therefore consistent with results from our pig and our rat studies, suggesting that the kinetics of siRNA biodistribution during EVHP is similar across species. To further identify cardiac cell types that take up DCA siRNA, we characterized siRNA internalization at the single cell level. Rat hearts were perfused for 6 hours with or without unconjugated or DCA conjugated siRNA, and we then performed flow cytometry on single-cell suspensions isolated from ventricular biopsies to identify populations that are Cy3 positive. Pictured here is the gating strategy used to identify each major cardiac population, with representative histograms which show the Cy3 fluorescence intensity shift across treatment conditions for each cell type, where we see that the DCA siRNA treatment in red enhances cellular uptake compared to unconjugated siRNA controls in blue or untreated hearts in gray. Quantification of Cy3 median fluorescence intensity reveals that DCA siRNA-treated hearts have greater Cy3 signal across all cardiac cell types, nearly 1.5 to 2-fold, and is especially enhanced in bulk immune cells and macrophages, more so than unconjugated siRNA in blue, suggesting that DCA is in fact driving the cellular internalization of siRNA during EVHP. Now that we've established that DCA conjugated siRNA drives cellular internalization in 3 species models of normothermic EVHP, we then wanted to assess the impact of siRNA dose on delivery. And so pictured here are representative fluorescence microscopy images showing Cy3 labeled DCA siRNA in red delivered to rat hearts during EVHP across a range of doses, including untreated hearts here on the left, then from siRNA doses from 0.1 micromolar to 2 micromolar, where we see a concentration-dependent increase in signal within the whole heart as the dose increases. Quantification of the mean fluorescence intensity of the heart ventricles also demonstrates this dose-dependent increase, with a 1 and 2 micromolar DCA siRNA group significantly enhancing MFI compared to untreated control EVHP perfused hearts. To further identify cardiac cell types that take up the DCA siRNA across this range of doses, we characterized siRNA internalization at the single cell level, again, using a similar flow cytometry strategy as before. And across this dose range from untreated to 2 micromolar, we see a positive shift with greater DCA siRNA concentrations administered during EVHP compared to untreated controls in gray. Quantification of Cy3 MFI demonstrates a dose-dependent increase in signal across all cardiac cell types and is significantly enhanced in hearts treated with the 2 micromolar DCA siRNA compared to other doses, suggesting that this 2 micromolar DCA siRNA is the optimal dose tested to enable robust delivery of siRNA within 6 hours of EVHP. The ultimate assessment of siRNA functionality is then whether we can achieve JAK1 target mRNA silencing. And so then compared to untreated hearts or hearts perfused with a DCA non-targeting siRNA control sequence, or NTC, the DCA JAK1 siRNA trends toward suppression of JAK1 mRNA expression levels at a 1 micromolar dose in 6 hours of EVHP. And we then assessed the impact of perfusion duration and siRNA dose on DCA siRNA-mediated JAK1 silencing, where we see significant silencing of our target after 14 hours of perfusion and with a 2 micromolar siRNA dose, suggesting that the DCA JAK1 siRNA can suppress further upregulation of JAK1 during EVHP. And importantly, untreated and DCA NTC heart show similar JAK1 expression levels, suggesting that JAK1 upregulation is due to the process of EVHP and not due to potential toxicity from the siRNA. As we move forward with leveraging EVHP as a platform for siRNA-based gene modulation, and now that we've assessed the impact of siRNA conjugate, dose, as well as perfusion duration, we are uniquely situated to advance this technology and investigate clinically relevant outcomes, including silencing several known IRI targets and ultimately evaluating organ function and post-transplant outcomes with these studies currently underway. Imagine a not-so-distant future where IRI can be targeted through a multiplex of cell type-specific siRNAs, such that those 10 to 30% of heart recipients no longer develop primary graft dysfunction, thus critically expanding the pool of available donor hearts for transplantation. So we believe our work lays the foundation for these future studies. In closing, I would like to thank everyone involved at MGH, UMass Chan, and Shriners Boston for really enabling this work, and I am incredibly grateful to everyone listed here, specifically bolded names, as they have helped me push this project forward immensely. And so thank you all so much for your time and attention, and I look forward to answering any questions. Thank you.
Julie, that was a fabulous presentation. Great job. We probably have time for one question. Dr. Billia?
Julie, fabulous job.
Thank you.
So your uptake of the silencer RNA is not really cell-specific, cell type-specific, and yet you talk about the world where you envision the cell specificity. So any thoughts about how you would be able to do that?
Yeah, that's a great point. I think, first off, we wanted to profile the DCA siRNA uptake across cell types in this particular setup for clinical application. And I think, the fact that we can hit a lot of the cardiac cell types is very promising because, depending on what target you're trying to go after and its expression levels, in the ex vivo period, in the post-transplant period, I think we have a real option given, for example, that we can really, get the siRNA into cell types like macrophages, like the general immune cells. This could allow you to kind of tailor your target selection in general. Our lab also has a variety of other conjugates that we can explore in this setup, but DCA siRNA is the most broad in terms of cardiac delivery.
So great talk. Thank you very much.
Thank you.
And in the interest of time, we'll move on to our second speaker, which it's my pleasure to introduce Pieterjan Kerckhof. Dr. Kerckhof is, let me find this here, is actually from Leuven, Belgium, and he will talk. So he is the 2023 Research in Immunology Professional Community Award winner for excellence, and he will talk about morphometric airway changes in explanted human lungs with chronic lung allograft dysfunction. All yours.
Great, thank you for the introduction and thank you for letting me be here. So I will indeed talk about chronic lung allograft dysfunction, or CLOT, or just simply said, rejection after lung transplantation. Oh, I'm sorry. I'm just trying to close this a bit. All right. Okay. So there are two main phenotypes of CLOT. We have our restrictive allograft syndrome, so the restrictive phenotype, and there's a lot of fibrosis, like you see on the left under side here. And then we have the second subtype, bronchiolitis obliterans syndrome, BOS, which is the obstructive subtype with a lot of obliterations in the bronchioli. Actually, we also have a third subtype, which is just the mixed, called the mixed subtype, and this is just a mixture of both of them. So the purpose of this talk is actually to know what happens in the airways of CLOT. And well, something like this, like we see here, does happen. All right, so for the data that we used. As controls, we use 6 explanted unused donor lungs, and we also use 6 BOS lungs, 3 mixed lungs, and 3 ROS lungs, and these are all end-stage explanted lungs. Here are some more details about these lungs. I will skip over this quite fast. Now for the methods. So we obtain first our lungs as inflated, frozen human explant lungs. We take a micro CT scan from these, and then we can visualize our entire airway tree actually. Then we also take tissue cores from this lung. And on these tissue cores, we again take a micro CT scan. We can again visualize the airways on this micro CT scan, but now into much greater detail. And now we can also, on these cores, visualize the terminal bronchioles. And we can also characterize and see all the lesions in these small airways. We can also integrate both together. So we begin with our airway tree. We divide this into several segments, and each time the airway bifurcates into two other airways, there is another segment. Schematically, this looks like this. So we first have our main stem bronchus. This divides into two other bronchi of generation 2 and so on. And in this analysis, all airways distal to any obstruction are not included. So let's talk about obstructions in the airways. Here we see the number of patent of open airways throughout the generations of each lung type. And we see in green and red for mixed and for BOS lungs that this is severely decreased. So there are a lot less open airways, and this is caused by obstructions. For us, this is quite comparable to the control, and only in the most distal airways around generation 15, there we see a decline. Now here is just depicted indeed over the generation, the number of obstructions, which is very similar to the previous graph. So I will skip over this quite quickly. And here we see the number of small bronchioles left over in the lung, small bronchioles which are still open. And we see that this is indeed also severely decreased in BOS and in mixed lungs, and less so in ROS. Here we only looked at bronchioles between 0.8 to 1.2 millimeters in diameter, and not smaller because of our resolution. But if you do want to look at smaller airways, like the terminal bronchioles, we need our tissue cores. And the next slide is about that. So here we see an example of BOS where we see the terminal bronchioles and the respiratory bronchioles, and they seem perfectly normal here, which is actually often the case in BOS. So the terminal bronchioles are quite safeguarded from most of the lesions. While when we look at ROS on the right-hand side, we see that, well, there's for sure something happening there in the airways. And this is quite typical as there exists a lot of fibrosis in ROS, and this fibrosis constricts the smallest airways and makes them actually obliterated often. And this is also why the previous graph you saw a decline in open airways in ROS, only in the most distal airways. So now I will show a bit all the different obstructions that are to be found in clot lungs. Just for starters, this is a normal lung, nothing to see, normal airway, nothing to see here. And here we see a really small lesion actually, sometimes something we sometimes see, and we call this a web. And this is very likely related to mucus. And the relevance is not very clear of this. This is again a web, a bit more severe and probably also related to mucus. Again, the relevance is not very clear, and this is to be studied further. And here we have then an example of obliterative bronchiolitis, where the airway lumen is completely obliterated. This can again be in part caused by mucus. From this CT image, we cannot be sure yet. And here we also have an example of constrictive bronchiolitis. And here we see on the right lower side that the airway lumen is narrowed, constricted. And then here we see something similar, which we call a vanishing airway, where the airway lumen is completely disappeared. So we have all these subtypes, and we do hypothesize that there's a temporal evolution. We cannot prove this yet. This is for further research. But the idea is that it starts with some inflammation in the airway wall. Then we get reactive mucus creation. And as this gets more severe, we get obliterative bronchiolitis. And in the end, collagen formation occurs with narrowing of the airway, and in the end, vanishing airway. So this is the hypothesis that we're going to test into the future. Now I have some images of the cores and the airways in our lung cores and what's all going on there. So here, this is a Bos core, so a piece of Bos lung, and in blue, this is the airway. And here we see a lot of small lesions. There we see a small web, and all of these lesions in this core actually are probably related to mucus. And again, this relevance of these small lesions is really not that clear. And it's really, we still need to investigate further. All right. Now, here we have an example again of a Bos core, and here we will see a vanishing airway. There is the airway, well, there should the airway be. And we see that there is just a small white stripe between those two pieces. And this is where the airway was, and this is the remainder of the airway wall. So indeed, the airway completely, well, the lumen is completely gone here. And then here is again one example of the core of the mixed phenotype. Here we see a lot more fibrosis, and there the vanishing airway, but we will go back to it. So there's an artery in fibrosis. And there now we see the airway, which will disappear. And around the airway, a bit more white, is the wall, and this also disappears completely. Indeed showing that this airway, because of the inflammation, well, does disappear completely. Right, and this is just an overview to show all lung types. And there we have some obstructions in all these clot lungs. Bronchiectasis is present mostly in Bos. And you see also in Bos that there are really a lower number of branches because of all these obstructions. All right. Now this is a typical view of a core in clot. And here we see the airway running through this core, and we also see those red pieces, and these are all obstructions in this core. And this is a really typical view. So I have these all these images here of all these different obstructions. And I will zoom in into some of these with histology, with the Masson-Strike-Chrome staining. And here on the upper part, we see that the airway lumen is severely well constricted. It's very, very small, as we see on the right upper side. And around it, there are a lot of lymphocytes. So this is some lymphocytic inflammation. And on the lower side, we see in the same airway just below some mucus in the airway lumen, with around also the lymphocytes and some formation of fibrosis, as we see in blue collagen. And here is an example of a vanishing airway. On the right-hand side, we see those red arrows, and these indicate smooth muscle cells. And this indicates that there used to be an airway there, but it has disappeared into, well, nothing, and some fibrosis has been left over in blue. This is the collagen again. And then here is another example of constrictive bronchiolitis. On the right-hand side here, we see the airway, which is smaller than it should be, and it's constricted, and all the epithelium is here gone. And there is a lot of blue, so a lot of formation of collagen here. And this will likely evolve, but this is a hypothesis still at the moment, but this will likely evolve to a fully vanished airway. All right, so now we have seen all these obstructions, but what does this mean for the lung, right? I will try to build this up. So for the first step is looking at this picture. Here we see two sister airways. One has a large diameter, the other one has a smaller diameter. And this is the real life situation in lung. If one branch like bifurcates, we often we do not get two perfectly similar branches, right? And I found that the branch with the smaller diameter was preferentially obstructed. And this is the graph of that, all right? So it seems that the smaller the diameter of the airway, the higher the risk of obstruction, all right? Then here we see the division of small airways throughout the generations in the lung, now only for control lungs, all right? Now, if we look at the clot lungs, we see that this division is very similar. This means that the generation itself has no effect on obstructions, right? Only, like we just previously saw, the diameter has an effect, but the generation does not. And this can be somewhat confusing. But if you go to the next slide, I have an example of this. So here we see an array of generation 8, 12, and 16. And well, these are then airways, as we see here, with a comparable diameter. So they also have comparable risk of getting obstructed. Now you would say, okay, only the diameter matters and not the location in the lung. But you do also see that this is the same distance from the terminal bronchioles. And actually our conclusion from this is that it is likely that the distance from the terminal bronchioles is dictating in the occurrence of obstructions, while the terminal bronchioles themselves are quite safeguarded from this. The hypothesis around this is that during lung transplantation, the bronchial circulation is sacrificed, and this is important for blood supply for the bronchi or bronchioli, while terminal bronchioles can possibly survive a bit better just on the pulmonary circulation. All right, and then what effect has this that mostly the smallest airways are first affected? Well, something like this. So the largest airways remain quite normal and open, and then the preferential flow occurs in only these airways or only this airway, and something like this happens in the end in clot lungs. So there's a ventilatory imbalance, and only one region is still perfused. In this case, or it can also be some regions. I have some quantitative proof of this, which I'm not showing at the moment, but this is how it looks then in a real life pulse lung. You only have one or a few long branches left, and all the rest is just obstructed. So it's really one or a few regions that stand in for the remainder of lung function at the end of clot. Now I validated this a bit more, and here we see an example of a computational fluid dynamics analysis. And this means that I modeled airflow throughout a Bos lung, and for now this is only one Bos lung. And here we see the pressure distribution through that lung during inspiration. And on the left upper lower side, we see yellow and green, and this means that the pressure there is lower. And this is actually the region where airflow is quite normal, and the only region in this lung, while all other parts of the lung have high pressures because there's barely any airflow left there. So indeed, this shows also in this one Bos lung for now, that there's only one region still functioning in this patient, this end-stage clot patient, while all other regions are not functioning at all anymore. So in conclusion, I want to say that there's a lot of heterogeneity in airway lesions in clots, and we want to find in the future how this evolves over time to understand more what's going on. These lesions occur mostly in Bos lungs, but also do exist in Ros lungs to a lesser extent. And we understand a bit more now how lung function decline originates in the lung with those regional imbalance in ventilation. Okay, thank you for listening, and I'm ready to answer some questions. All right.
Thank you, Peter, and that's wonderful. We have time for a couple questions. Why don't we start off with Eric? And for the rest of you, can raise your hand since we are such a small group.
Yeah, that was a great talk. I'm wondering, in terms of scalability, is this technology, for early detection, and have you used it in, I guess, normal, not end stage, but sort of very, very early to see whether or not it could detect the onset of BOS prior to PFTs or something like that?
Yeah, that's a good question indeed. At the moment, we have not done this, so we only included end-stage lungs to see what's going on there, because it's also the lungs you can obtain. It's difficult to obtain a lung that's not end-stage, of course. The plan is indeed to have more insights in the previous stage, but this is for the future and a bit difficult. We do have in vivo scans of the patients, right? In vivo CT scans, but these are the resolution of these is a lot less good than these we have from the ex vivo lungs. And on these, you cannot see the small airways. So on these, you cannot learn a lot. Yeah.
If you had a transbronchial biopsy, would that be sufficient tissue, or do you have to have that kind of core thingy to do it?
Yeah, no, that would not be sufficient, because with the methods I just described, we first have our whole lung, and then we take a lot of cores from this lung, and then we select some interesting cores from that. So how we do that, we check a lot of cores, and then in these cores we look for the lesions where something is interesting, where something is going on, to investigate this further. And then we can slice this, we can do histology, we can do everything. We're going to do some transcriptomics also on that. But the main advantage of this approach is that we can really pinpoint all these lesions. So we just take what is the word? Transbronchial biopsy. You will probably not have a region that you can really say much about on this one biopsy. No.
Okay, thank you very much. Any other questions from our group that's online with us? Okay, why don't we move to the third talk then? Eric, I'll let you to introduce.
Yeah, so this will be Dr. Ko from the University of Tokyo, who is the Phillips Caves Award winner. He is actually in the OR, and so sent us a recording so that we can appreciate his great work.
So now we
I think it's muted.
Hello, everybody. My name is Toshiyuki Ko from the University of Tokyo Department of Cardiovascular Medicine. I'm very happy to have a chance to introduce my recent work in this web seminar. Well, the recent advances of the medical therapy for heart failure has substantially improved the patient's prognosis. However, the therapeutic response of the heart failure is highly heterogeneous among different patients. The patients who achieved left ventricular reverse remodeling, so-called AVRR, are known to have a good prognosis. Although many efforts have been made to predict AVRR so far, their prediction performance is still questionable. More importantly, none of them has been tested to be universally applicable for various etiologies of heart failure. As we know, the heart failure has an extremely large variety of its underlying etiologies. Such variety make it very difficult to develop a universal biomarker for the prognostic prediction. So we try to approach this problem with the insights from our basic research. In recent years, single-cell RNA sequencing, which enable us to know the whole gene expression profile of each individual cell, has totally changed the research of molecular biology. And I have performed various studies using single-cell analysis for both human and murine heart. For example, in the past 2 studies, we have isolated cardiomyocytes from both healthy and failing hearts and performed single-cell RNA-seq analysis to investigate its transcriptional heterogeneity. As a result, we found a part of cardiomyocytes from DCM patients are characterized by high expression level of DNA damage response-associated genes. In addition, interestingly, the expression level of these genes is inversely correlated with the therapeutic responsibility of the patients. So based on these findings, a hypothesis came up that if we can evaluate DNA damage in heart tissue, it could be useful for the prognostic prediction. So to test this hypothesis, at first I did a pilot study. I collected the endomyocardial biopsy specimens from patients with DCM at the time before the introduction of medical therapy and performed DNA damage marker staining. As a result, I found that the patients with poor prognosis had a high level of DNA damage accumulation in their heart tissue, reviewed by 2 biomarkers, poly-ATP ribose and gamma-SAX. So I started this multi-center study. We collected 175 biopsy specimens of HFrEF patients with multiple underlying etiologies. And I performed immunostaining of the DNA damage markers and quantification of them and evaluated its predict performance for AVRR. For this study, we developed an AI algorithm which can quantify the fluorescence intensity of the staining signal in each nucleus and automatically set the threshold to count the positively stained nuclei based on the histogram of the fluorescence intensity. So the software automatically calculated the number of DNA damage marker positive nuclei for each sample by using the cell algorithm. The DNA damage marker staining positivity was calculated like this. So this is the patient characteristics. Among 175 HFrEF patients, 55% achieved ABR at 1 year treatment. As shown in this table, they had a variety of underlying diseases. And consistent with the previous studies of ABR prediction, the patients with ABR had a shorter disease duration, the less CLBBB, and the smaller AV chamber geometry size as a baseline. And at the time of ABR assessment, the medical treatment was fairly comparable between the two groups. After quantification, both DNA damage markers were highly positive in those patients without ABR, and the ROC curves presented with very high performance. So both DNA damage markers can significantly predict ABR even after IPW analysis to balance the various baseline characteristics shown here. So when we classify the patients with two groups by the cutoffs of the DNA damage marker staining positivity shown here, it successfully predicts the composite hard endpoints. The hazard ratios, every 10% increase of the staining markers were approximately 1.5 times higher, both before and after IPW analysis. By weighted linear regression model, we observed a beautiful and remarkable reverse linear relationships between the stained DNA damage marker positivity and the probability of ABR among various etiologies, represented by the dots in different colors. So both we and the other group have previously reported the assessment of the genomic mutation can predict the prognosis of DCM patients. To date, the sarcomeric gene mutation is known to have a better prognosis than non-sarcomeric gene mutation. So within the cohort, we had approach to the genetic information of 89 patients with DCM. Among them, 32 patients carry the pathogenic or likely pathogenic variants in cardiomyopathy-related genes. We found that the patients who had sarcomeric gene mutations had less DNA damage compared with patients with non-sarcomeric gene mutations. So the information of myocardial DNA damage is independent, significantly predictor for the prognosis. And the combination of DNA damage marker staining and the genetic information only improves the prediction performance a little bit. So the association between the genotype and the treatment response in patients with DCM was partially mediated by the myocardial DNA damage. As we know, the precision medicine in cancer therapy has already become the clinical standard. In this study, we demonstrated that the evaluation of DNA damage in the heart tissue will be a promising approach for the risk stratification and the decision-making of the treatment plan. So recently, more and more evidences suggested that DNA damage is associated with pathogenesis of various cardiac diseases, such as AF and the aging of the heart and the HFpEF. So also in this study, we only studied the DNA damage in the various etiologies of HFrEF. The same approach has the potential to be extended to the HFpEF and other cardiac phenotypes. So taken together with our previous and the current results, we conclude that the evaluation of DNA damage is useful for the prognostic prediction of healthy patients, regardless of its underlying etiologies, thus opening the door of the precision medicine in the field of heart failure therapy. And we are currently working on several new translational projects relating to the present study. First, in order to non-invasively evaluate the DNA damage in the heart tissue, we are now trying to develop a nuclear medicine assay that can specifically detect the DNA damage. Second, the DNA damage is actually not only a simple biomarker of the heart failure, but also a therapeutic target of the heart failure. We have performed the compound screening using the iPS cardiomyocytes from the patients with DCM and found that the vitamin D2 can safely and significantly reduce the DNA damage in the cardiomyocyte. So finally, I'd like to thank all my research collaborators and my colleagues. And thank you so much for your kind attention.
So I want to thank Dr. Ko, even though he's absent, for this incredibly well-given taping. I'm sure that if anybody had questions and wishes to reach out to him, he would be more than happy to answer your questions. It's unfortunate that he is trapped in the OR, as our surgeons are. We completely understand that. And what I'm going to do is I'd like to close. We're a few minutes early, but I wanted to close our webinar. Thank you to Eric as well for co-moderating with me. And also to remind you about ISHLT happening in Boston next year, a little bit different date towards the end of the month, starting on a Sunday, so slightly different version of what's happening. And that also to remind you that our next PC webinar is actually a pharmacy one that is actually happening on November the 19th. So not too far distant future, just 2 weeks away, and hope to see people there. And again, thank you for your attendance. It was incredible to hear these really thoughtful talks and to see how our early career folks are really pushing the boundaries and keeping us on our toes. So with that, I'd like to end our webinar for today. Thank you.